Okay, so let's start. Welcome everybody to today's VTLS seminar. Our speaker today is Jonathan Auguste, and he received his bachelor degree and his PhD from the University of West Indies in Trinidad. His bachelor was in the field of biochemistry and PhD in molecular genetics. He has been working extensively in arboviruses, and is an area where my knowledge is very little, so I'm taking full advantage of this seminar to educate myself. He continued his work as postdoctoral fellow in the Department of Microbiology and Immunology at the University of Texas Medical Branch in Galveston, after which he joined the rank of assistant professor of arbovirology in the Department of Microbiology and Immunology in Galveston, Texas as well, before moving for his permanent position at Virginia Tech in the Department of Entomology. I have to say that Jonathan has received several awards that are worth to mention, in particular the NIH K22 Career Development Award. He, I don't know why I'm reading without my glasses. He also received the Robert Shop International Fellowship in Infectious Diseases. He has also received an award from the American Society of Microbiology that unfortunately he needed to decline because he got too many awards. He couldn't get all at the same time. Jonathan works on arbovirus ecology, also in emergence, evolution, and spread of arboviruses, and the pathogenesis. He's gonna give us kind of a really big background of what is going on right now in his lab, where everything is going, and his latest publications. He currently has support from very many agencies, and including NIH, from the NIAID, and also from several seed grants in our university. Jonathan, without further ado, the audience is yours. Thanks a lot for joining us today.
Thank you, Carla. I'm just going to go ahead and cancel my video for now. I'll reopen it when we take questions later on. I don't want you guys to see me looking between my screens that I have up here.
And thanks very much for wearing a tie.
No problem. All right. So again, thanks, Carla. I do appreciate the opportunity to talk about what my lab has been doing recently. So thanks again. Just one quick note to those in the audience. My full name is Albert Jonathan Auguste. I do go by Jonathan. But if you have a PubMed Jonathan Auguste, that's not me. So you actually need to look at Albert Auguste. Otherwise, you'll think I've never published. So I'm a relatively new assistant professor in the Department of Entomology, and I'm housed in Herein Hall. I started in August 2017. So I'm going to be talking about what we've been doing in the past 2 or 3 years at Virginia Tech today. Tell you a little bit about what we've been doing in my lab. So there we go. So first, I'd like to thank the people that are actually doing the work and the people that have been supporting me throughout this research project here at Virginia Tech. So I'd first like to thank my lab, specifically my graduate students, one undergraduate, and my lab manager. So I have about 5 or 6 people in the lab currently. The project, the data that I'm presenting today is going to be was derived from 3 people primarily. Ms. Danielle Poirier, who is my graduate student, and this is a dissertation project. Ms. Sarah Wilson, who was an undergraduate researcher in my lab. She has now left us and gone to UGA, where she's doing biomedical engineering, a master's program, and Mrs. Dawn Auguste, who is the research scientist in the lab and sort of holds the lab together in my absence. There's also a lot of people at Virginia Tech that I'd like to thank. This has been an especially collaborative project, and I'm very excited about the collaborations that we've set up throughout this project. So I'd like to thank Dr. Extra Meng, who's really been my mentor here at Virginia Tech and our sounding board for my research projects. Dr. Coy Allen, who's my immunologist and has really been instrumental in the completion of several of these studies. Dr. Clayton Caswell, who's willing to house one of my students for some protein expression assays that we've done. Dr. Powell Michalak and Dr. Lincoln, who assisted with differentially expressed gene studies that we've done. Dr. Joseph Basagani Herrera and Dr. Andrew Lieber, who are both associated with Virginia Tech, but they have a company called Landos Biopharma out of the Corporate Research Center, and they've helped with a lot of the immunology studies that we've done. Also, for some folks, more folks in VetMed, Dr. Nisha Dugal, who's willing to provide viruses and share some of her insight into in utero transmission studies with Zika, and Dr. James Ruega Lucarelli, who assisted in the development of the first clone for these viruses. As you mentioned, there's a lot of funding that I needed to acknowledge, including some awards from the NIH, including my K22 and R01 awards. So viruses can emerge in humans and animals for a variety of reasons. I just took the liberty of categorizing these into 4 main categories, being those associated with the vector, those associated with the environment, those that may be associated with the virus itself, and those that may be associated with human action. So for example, if there's a significant increase in vector population size or significant increase in the geographic distribution, where they are now becoming into contact with susceptible hosts, that can often lead to viral emergence. Another really good example with respect to the environment is that climate change is now causing increasing temperatures. This increasing temperatures is also facilitating the spread in geographic distribution of the associated vectors. That again can lead to virus emergence in humans and animals. Often, but very rarely, the virus itself can actually adapt to increased transmissibility or for increased host susceptibility, and that often leads to virus emergence into new systems. Finally, there are factors related to human actions that can also directly or indirectly lead to emergence. For example, significant reductions in pre-existing immunity, which is often a result of reduced vaccine availability, can lead to virus emergence. A perfect example of that is yellow fever virus, where there's a really good vaccine available, but it's no longer available in some places globally, and that has allowed the virus to re-emerge in places of South America and Africa. So, unfortunately, for my lab members, my research interests are quite broad. And we are interested, and we do research in all 4 of these categories. We are interested in arboviral surveillance, and we also perform vector competence studies. My lab is focusing on new strategies for reducing vector competence of mosquitoes for arboviruses. My lab is also interested in understanding arbovirus transmission cycles in nature and developing new animal models to better inform vaccine testing. Currently, my lab actually works with mice, rats, hamsters. We've just expanded to poultry, including chickens, ducklings, and quail. And next year, we're actually moving into sheep, which is pretty interesting. And we have some collaborators we work with for non-human primates. We also work with virus-host interactions, where we try to understand the correlates of arboviral pathogenesis as it relates to vaccine efficacy. But today, I'm going to be telling you a little bit more about our research into vaccine development that we've been doing here at Tech. So, why vaccines? When SARS coronavirus 2 emerged, that's the virus that causes COVID-19, about a year ago, there was about 130 new vaccines and preclinical testing within 1 year. So why is there still a need for new vaccine strategies? Because ultimately, vaccine development is plagued by trade-offs, so that when vaccines are exceptionally safe, they are not often immunogenic or as effective as some of their other counterparts. A good example of this is a kill vaccine. It's extremely safe, but it's not as effective as some of the other vaccines that may be available. On the other hand, we can have vaccines that are exceptionally immunogenic and effective, but don't often reflect the same safety profiles as you might have with killed vaccines. And a good example of that is a live attenuated vaccine, where the virus can actually replicate in the host. So as scientists and as researchers, there's a dire need to always explore new strategies where we can achieve a better balance between vaccine safety and vaccine efficacy. So I was interested in developing a new vaccine for flaviviruses. And to tell you about the new strategy that we developed, I need to tell you a little bit about insect-specific viruses. So insect-specific viruses are host restricted for replication in insects only. So a typical arbovirus is transmitted between a competent vector and a susceptible vertebrate host. Often that can result in spillover infections into animals and humans. However, insect-specific viruses are unable to replicate in vertebrates and is therefore transmitted between mosquitoes only. It's likely maintained in nature by vertical transmission, horizontal transmission, or venereal transmission among mosquitoes. So I was interested in using these viruses as a platform or delivery vehicle for flavivirus vaccines. So to do this, I put on my virus hunter hat for a little bit, and I started screening mosquitoes that I collected in Trinidad and Tobago during my PhD research. It didn't take long. These viruses are quite predominant, quite prevalent in nature, and we isolated several new viruses that we went on to later characterize and publish on. However, we were really lucky in that we actually were able to isolate a novel insect-specific flavivirus, which is in the same genus of virus that I was hoping to develop vaccines for. We call this virus Aripo virus. Aripo virus was isolated from a pool of Saurophora albipes mosquitoes and was first detected because of the massive cytopathic effects caused in mosquito cells in cell culture. This is a rare and unique phenotype. Other pathogenic flaviviruses don't actually cause this level of cytopathic effects. So that was a unique trait to Aripo virus. We sequenced Aripo virus, the complete genome, and we built a maximum likelihood phylogeny. And what we were able to show is that Aripo virus groups together with other insect-specific flaviviruses, but it groups together with these viruses within the main group of pathogenic mosquito-borne viruses like dengue, yellow fever, Zika, West Nile. And this was particularly interesting and very important because there's another group of insect-specific flaviviruses that are very genetically divergent. So what this phylogeny suggests is that Aripo virus shares a closer evolutionary relationship with the pathogenic flaviviruses. And Aripo virus, like other insect-specific viruses, does not cause any CPE in vertebrate cells. It did not cause any disease if you actually put the virus directly into the brain of suckling mice. We saw no evidence of replication after infection of vertebrate cells. So I'll just go through one simple graph that we have here that sort of shows this. When you infect mosquito cells, you can see a nice replication of Aripo virus. But once you start infecting vertebrate cells, the virus degrades over time and it just disappears from the cell culture supernatant. So we were very happy. We isolated a virus that we believed might have been a good vector. So we started exploring some of the phenotypic characteristics associated with this virus. The first thing that we discovered, which was very interesting because previous studies suggest that insect-specific viruses are host restricted because they are unable to enter vertebrate cells and they are unable to replicate or translate proteins even if the genome is inserted directly into vertebrate cells. Well, what we were able to show very early on is by using a transmission electron microscopy experiment when we infected vertebrate cells, we were able to show that the virus actually does become associated with cell membranes and enters cells via a process called clathrin-mediated endocytosis. This is receptor-mediated. And you can see this sort of like pit that it forms. That's a clathrin pit based on the double membrane structure, the spikes that we see here. And you can actually see the virus being endocytosed within a cell within 2 to 5 minutes after infection. And this was particularly interesting because this now tells us that this virus might have actually been able to enter cells and might therefore already be immunogenic, which is what we looked at next. So to see how immunogenic this virus might be, or if it is immunogenic, we infected murine macrophages. And we looked for innate immune responses to infection. And we did this using bioplex assays, so looking at proteins, secreted cytokines, and the cell culture supernatant. And after infection, we were able to clearly show that Aripovirus induced strong inflammatory and pro-inflammatory cytokine responses post-infection, as early as 6 hours post-infection. So this was very interesting. What we had is we now discovered a virus that was already immunogenic, which will contribute substantially to our vaccine development efforts. We wanted to explore this phenotype a little further. So we did an experiment to see whether or not we can recapitulate that immunomodulatory effects in mice rather than in cell culture only. So to do this experiment, what we did was we infected CD1 mice. Now, CD1 mice are immune competent mice. So you see relatively little disease unless the virus is very pathogenic for this strain of mouse. And we infected CD1 mice with Aripovirus 7 days before West Nile, co-infected with West Nile, and 1 day before West Nile. You can go ahead and ignore the controls for now. What we were able to see with this model is that mice that received Aripo 7 days prior to West Nile infection mostly succumbed to illness and died by 15 days post-infection, and they showed significant weight change. Mice that were co-infected with West Nile showed reduced mortality, but also showed weight loss. However, those mice that received Aripo 1 day prior to West Nile all survived and showed no weight loss whatsoever. So this sort of alluded us to hypothesize that Aripovirus does likely produce a strong innate immune response, which may have offered this protection. So we studied this further, and then we used IFNAR receptor knockout mice. So this is a type 1 interferon receptor knockout mice, so it's efficient in interferon alpha and beta, and what we believe is likely the factor that produces this sort of cross-protection effect we see. And we show that Aripo is safe. If you just put Aripo only, it's safe in these mice. But if you put Aripovirus 1 day prior to West Nile, these mice do lose weight, and they do die at the same rate as West Nile virus only infected mice. So this sort of like hints to the fact that this protection we saw was because of a type 1 interferon immune response. So we had a very good platform that we believe we can use to develop our chimeric vaccines. So the approach we used was to develop a chimera where what we do is we would substitute the structural proteins of Aripovirus with that of Zika virus. So the structural proteins are the proteins that make a virus look the way it is. It's the surface proteins. So by substituting this with the Zika virus structural proteins into the genome, we now have a virus that looks identical to Zika on the outside, but contains the replicational defective genome of Aripovirus on the inside. So it looks identical to Zika, but still can't replicate in a vertebrate cell because of the Aripo genome. This was actually developed by James Weger, who's from the College of Vet Med. So why Zika virus? I usually say it's not because NIH dedicated millions of dollars to Zika virus research. There's actually a lot of other factors that went into place when selecting Zika virus. Some of the key factors were that Zika actually has very reliable murine models. It's a BSL-2 pathogen, which allows us to work with it in our common lab, and we don't require biosafety level 3 facilities. And there are a significant number of other Zika virus vaccines that we can actually directly compare our platform against. But more so, Zika virus is a very important flavivirus for human health. It is distributed nearly globally and can be transmitted by multiple routes. Not only a mosquito, not only through the bite of an infected mosquito. It can also be transmitted through sex. There's a possibility that it can be transmitted through blood transfusion, but it is known to be transmitted from mother to fetus via in utero transmission. Zika virus disease can manifest in a variety of ways, but it is most known for its congenital birth defects, including microcephaly, which is a narrowing of the head circumference, and brain development issues, brain tissue development issues. So we had our chimera that we developed. We knew that it was likely going to be immunogenic, but we needed to evaluate first whether or not this was going to be safe after this substitution was made. So our hypothesis was that immunization with Aripo Zika is safe as it retains Aripo's natural host restriction in vertebrate cells. So we were able to rescue this chimeric virus. Initial rescue titers were relatively low. So we knew this is an insect-specific virus, so if you keep passaging it in C636 cells, it will likely adapt for its natural replication potential, and it did so within 5 passages. We continued to passage this for a maximum of 14 times to allow this any adaptive mutations to become fixed to the population. Here we can see the growth of Aripo virus and the growth of Aripo Zika virus, the vaccine, over time, over the course of 7 days. And you can see that they sort of mirror each other in its growth kinetics, and actually far out replicates Zika virus the wild type Zika virus. So we knew we were able to rescue our virus, and it grew really well in C636 cells, but we wanted now to confirm that its replication defective in vertebrate cells. So we did an experiment where we looked at replication both within cells and in the cellular supernatant fraction after infection. And to do this, we used both the Aripo backbone, we used the Aripo Zika vaccine and Zika virus as a control. And what you can see here is that when you look at the extracellular fraction, the cell culture supernatant after infection, you can see that the virus, again, after infection, just tapers off over time and degrades over time. In contrast to Zika virus, which is replication competent, it grows really well and is secreted from cells. To confirm that there was no replication happening within the cell, but that there was defective particles being produced, not defective particles being produced, we looked at intracellular replication, and again, we saw only a decline in virus over time within the cells. So this is going slow. All right. So we knew that our virus was replicating and that it produces Zika proteins by Western blots in insect cells. So we used protein expression as a surrogate to investigate virus replication in a variety of vertebrate cell lines. We selected cell lines that were particularly susceptible to Zika virus replication, including conventional cell lines we use in our lab, like Veros and African green monkey kidney cells, hamster cells, human cells. But we went one step further, and we selected cells such as placental cells and testicle cells that are known to actually allow for the persistence of Zika virus. And what we did is we infected these cells at a relatively high MOI, and we measured protein expression using anti-Zika envelope antibodies. And what we were able to show is that we could not detect Aripo Zika proteins in any of the vertebrate cell lines, in contrast to the C636 cells, where expression was far greater than the Zika virus infected controls. So because we used a flow cytometric approach, we were actually able to take snapshots of each cell as it was counted during sampling. And this is just sort of an overview of like the data that we collected. And you can see that Aripo Zika, there's, again, from the quantitative data, there's no expression except in insect cells, but Zika, you can see clear protein expression in vertebrate cells. Because we're working with RNA viruses, which have a slightly higher evolutionary rate than other viruses, we actually wanted to confirm that our virus could not adapt and gain the ability to replicate in vertebrate cells. So to do this, we actually did a blind serial passaging experiment where we infected cells, Vero cells, and BHC cells with Aripo Zika and other controls to see if the virus was going to be able to gain that replication function. So we inoculated an extremely high MOI in our very first passage of about 1,000 or 10,000. And then we did not wash these cells post-infection. So we left that virus on there. Then we used, 5 days later, we would take an aliquot from that and passage it into the passage 2. At this point, we started washing cells after infection to remove residual virus. And what we can see is that time point we take 5 days after infection, you can see Aripo Zika is present because we didn't remove the high virus titer that we actually put onto cells. In the second passage, there was some residual virus left over. But at the second passage where we began washing residual virus off, we lost Aripo Zika in the later passages. So it suggests that the virus wasn't able to adapt and gain that function because it was lost to the blind passaging. So that was some really interesting in vitro studies that suggested that the virus was safe. So we then started looking at in vivo. And the first study we did is we explored the physiological effects of Aripo Zika immunization in contrast to Zika and the backbone in wild-type mice. So we vaccinated 4-week-old wild-type mice and we performed CBC studies. And what we were looking for is any sort of effects that Aripo Zika might have that might be comparable to other flaviviruses. We were primarily looking for things like thrombocytopenia associated with dengue. And what we saw is that there's almost no physiological effects except for the mean corpuscular volume and the red blood cell width. And my pathologist says that by themselves, this doesn't really allude to any pathological disease that we are aware of. So then we moved to the most susceptible model of arbovirus replication that's available to us. And we inoculated Aripo Zika directly into the brains of 2-day-old suckling mice. Here we used a variety of controls, including the Aripo backbone. Zika is a positive control to show classical arbovirus disease, but we also included a gamma-irradiated Zika, which should reflect what we see with Aripo Zika, because this is an inactivated Zika virus control. So what we were able to see here is that Aripo Zika mice that were intracranially inoculated gained weight at the same rate as our PBS controls and the Aripo controls. And none of these mice succumbed to any illness or death. In contrast, Zika virus, as expected, killed all mice within 7 days, and there was a significant weight loss after 5 days post-infection. Particularly interesting, given the extremely high dose of Aripo Zika we inoculated into mice, there was absolutely no histopathology associated with Aripo Zika, in contrast to the inflammation that we saw with Zika virus throughout brain sections. So in summary, both Aripo and Aripo Zika show no evidence of replication in cell cultures, and this was evidenced by no replication kinetics as well as no protein expression. There was no evidence for gain of replication function either, and there was no detectable virulence in any of the murine models that we used to assess safety. So we now knew that the vaccine was extremely safe. We wanted to evaluate whether or not it was going to be as effective as some of the other Zika virus vaccines that's currently available. So our hypothesis is that immunization with Aripo Zika completely protects immune-competent and immune-compromised mice from all Zika-induced morbidity and mortality. So to do this, I'm going to just go through very slowly the study design we used for majority of these studies. But what I want to really go through here is the groups that we use. So PBS mice, mice that have received PBS vaccination and a PBS challenge. So these are healthy mice. Aripo is our backbone that we were exploring to see that to confirm that there was no sort of cross-protective effects as a result of just using the backbone. Aripo Zika is our vaccination group. We use the Zika PRV ABC59 control. This is a mouse attenuated strain, which isn't usually lethal for adult mice. And our sham group, these are PBS vaccinated Zika challenged mice. So these are the mice that will become sick and die, and that we can compare any of the other groups' lethality with. We also use the inactivated Zika, the gamma irradiated Zika virus. That's inactivated and should closely reflect what our virus should look like, because our virus should behave somewhat like a killed vaccine. So as for the study design, we would vaccinate our mice and we would bleed weekly to measure neutralizing antibody responses. After 1 month in a short-term study, we would challenge mice with a lethal dose of Zika. IFNA mice, again, these are the type 1 interferon receptor knockout mice. These are particularly susceptible to Zika disease, and they die within 7 days post-infection. After challenge, we would take blood samples to measure viremia, and we would monitor for weight loss and death over 14 days. What we saw is that Aripo Zika was exceptionally immunogenic in this mouse model, producing PRNT80 titers of about 2.5 logs within 7 days post-infection. This further increased to nearly 3 logs by about 1 month post-immunization, which is also comparable to our live attenuated vaccine surrogate that we use here. There was no neutralizing antibodies detected in any of the other virus groups that we used, such as the gamma-irradiated or the Aripo virus controls. So, as expected, given these robust neutralizing antibody titers, and for those of you that don't know, neutralizing antibodies, those are the antibodies that are able to actually bind the viruses and prevent infection. So those are the antibodies that protect you once you are exposed to Zika virus. As expected, given those neutralizing antibody titers, Aripo Zika immunized mice were completely protected from mortality. All our mice survived infection, in contrast to the Aripo group and the sham and gamma irradiated groups, which all succumbed to illness. Aripo Zika vaccinated mice also lost no weight and produced no virus in the blood. And that's particularly important because in the absence of detectable virus in the blood, that means a human cannot perpetuate transmission. In contrast to some of the others where we used our sham vaccinated group or the Aripo backbone, we saw significant viremia and significant weight loss. So that was the immune compromised model. And that model is sort of prone to higher neutralizing antibody responses post-immunization because of its immune compromised status. So we needed to confirm that the vaccine is, again, effective, but in an immune competent model. So using the same virus groups and the same experimental study design, we explored this in C57 Black 6 mice. However, because these mice are resistant to Zika virus infection, what we need to do to make them susceptible prior to challenge is actually administer an anti-interferon antibody. It's called an anti-interferon blockade to make that virus then susceptible, to make that mouse susceptible to Zika virus challenge. So we immunize mice in its fully immune competent state, and we allow that to go for the 28 days. Then we administer the anti-interferon blockade and challenge mice, and then we monitor for disease as before. What we saw here, which was extremely interesting, is that within 7 days post-vaccination, again, the Aripo Zika group produced robust neutralizing antibody titers, even more so than the Zika live attenuated vaccine surrogate that we used. At 28 days post-vaccination, this increased, and again, was still higher than our live attenuated vaccine surrogate that we used. Given these high neutralizing antibody titers, we again expected complete protection from disease and death, which is what we observed. Aripo Zika immunized mice were completely protected. There was no mortality, there was no weight loss, and no viremia was detected in any of the mice immunized with Aripo Zika. This was in stark contrast to the sham vaccinated and the gamma irradiated groups, control groups. So one of the important areas of Zika virus research, and one essential component for a good Zika virus vaccine, is the ability to prevent in utero transmission of Zika virus from mother to fetus. So we set up a study where we only compared Aripo Zika against PBS and sham vaccinated mice. And this study design is set up where we vaccinate mice, we bleed again weekly for 28 days. At 1 month post-vaccination, we mate mice, and we look for a vaginal plug after mating. Once you detect a vaginal plug, we call that embryonic day 0.5. And you let those mice go for 10 days, after which you can challenge with Zika virus. We take 1 blood sample after challenge to make sure, to be able to measure viremias, to make sure dams were infected. And then we monitor for weight loss and disease and death. And 6 days after challenge, we euthanize, and then we necropsy these animals to collect tissues for analysis. And what we were able to show here is that, as expected, Aripo Zika produced nice, robust neutralizing antibody titers. Aripo Zika actually protected all moms from weight loss. There's a nice weight gain in Aripo Zika groups. Aripo Zika also protected moms from viremia. In contrast to the sham vaccinated groups, where produced extensive viremia and there was weight loss post-challenge. But with respect to in utero transmission, Aripo Zika actually protected pups from weight loss. You can see that the sham vaccinated groups lost weight compared to the Aripo Zika and the PBS controls. And when you look at the tissues, being maternal, placentas, and spleen, again, you can see Aripo Zika completely protected against any virus presence in those tissues. When you actually looked now at, when we looked at the pups themselves, to look at virus transmission from the mother to the fetus and looked at the brain tissues of those pups, we actually saw lots of Zika in a sham group, but again, the Aripo Zika group was completely protected from any virus. So we showed that our vaccine was effective at short-term studies, but what we wanted to do next was to see if immunization afforded long-term immunity. As we all know, immunity wanes over time, so this is something that was especially important. So we vaccinated mice using the same study design, and 1 year later, we decided to do the challenge. And we used the C57 Black 6 mice in this group, so we followed up with an anti-interferon blockade. And what we were able to show is that even 1 year after infection, Aripo Zika immunized mice offered complete protection from any death, from weight loss, and from any virus, viremia, in these animals, in contrast to the Zika virus infected, the sham vaccinated Zika virus infected group, where we detected a lot of virus, weight loss, and death. We did, however, see that antibody titers did wane over time from about 3 logs to about 1 log. So that was interesting, although we did see complete protection. So in summary of that, Aripo Zika is highly immunogenic and produces a really robust neutralizing antibody response, and this is essential for all arbovirus vaccines. Aripo Zika immunization completely protects against all Zika-induced morbidity, mortality, and in utero transmission, up to 1 year post-immunization, and likely within 7 days of immunization. And a really key component is that this vaccine is actually able to produce all of that with 1 single dose. So we quickly wanted to evaluate what was sort of the mechanism underlying this robust protection that we observed. And to do this, we hypothesized that immunization is highly immunogenic and develops a robust T-cell response. So I'll go through this very quickly. And what we saw here is that we vaccinated mice, and 8 days post-infection, or post-immunization, we collected blood and spleen samples, and 35 days later, we collected spleen samples. What we wanted to use these samples for was to measure the cell-mediated response to immunization in these animals. Again, using the same virus groups, we measured total as well as specific T-cell subtypes. And what we saw is that in general, we did not see a significant increase in total CD4 or CD8 cells, but we saw increases in activated CD4 and CD8 cells, both within the blood samples and post-stimulation. So the next study that we did is, upon harvesting spleens, we purified splenocytes, and we stimulated these splenocytes with live Zika virus or Zika virus E peptides, and we looked at the response of the T-cell. We looked at the T-cell responses. And what we were able to see is that the T-cell responses were primarily driven by Th1 and Th2 responses. In combination with this, in addition to looking at the cellular response, we were also looking at, the cytokine expression profile post stimulation of these splenocyte cells. And what we were able to see is that we did see robust expression of IL-2, interferon gamma, and TNF-alpha. These are some of the common cytokines used in Flavivirus biology to understand inflammation. And again, with the Aripo Zika groups, whether or not it was stimulated with live Zika or Zika E peptide, we did see robust stimulation and production of cytokines at 8 days and 35 days post-immunization. So this is where things started becoming very interesting. We had a very safe, we had a very effective, we had a very immunogenic vaccine. But now we wanted to know, why is this vaccine potentially so immunogenic? Because it should theoretically behave like a kill vaccine or pseudo-inactivated vaccine. So what we did is we actually infected murine macrophages with these virus groups, Aripo Zika, Zika, control, which is mock infected, and we used a UV inactivated Aripo Zika group. And after infection with these cells, at 6 hours post-infection, we extracted RNA from these cells to look for differentially expressed genes. And we had a lot of help here from Dr. Ling Kang and Dr. Powell Michalak. And what we were able to show is that Aripo Zika actually has a sort of minimal effect in its regulation of genes, in contrast to Zika, which had a significant number of up and down regulated genes, and Aripo, which also had a significant number of up and down regulated genes. Also particularly interesting here is that Aripo Zika was very comparable to Aripo Zika UV inactivated groups, and that there's no difference, which sort of suggests that there's no replication, there's no underlying replication that might be going on that we were not able to detect. So we sent this data to Dr. Coy Allen, our immunologist, who inserted this information into IPA to look at what cellular pathways were up and down regulated post-infection. And he was able to show that there are numerous pathways associated with both innate immune responses and adaptive immune responses to immunization, all via this signaling hub of TNF-alpha, interferon gamma, and IL-2, which again, as I mentioned before, we were able to observe in our splenocyte stimulation studies. So we were able to identify that there were many pathways, again, associated with innate and adaptive immune responses that were related to those biological functions. So that was particularly interesting. We now actually going into this data and sort of exploring the molecular mechanisms or those cellular factors that might be producing the robust immune responses that we're seeing. All right. So in summary, Aripovirus chimeras are very safe. They retain Aripovirus's natural host restriction. Immunization produces robust neutralizing antibody titers as early as 7 days. So people might be protected within 7 days if it's as immunogenic in humans as it is in mice. It produces robust T-cell immune responses as well. Aripo Zika immunization completely protects against all Zika virus-induced morbidity and mortality, as well as protects against in utero transmission. And we're seeing that this might be a response of robust PRR signaling after Aripo Zika exposure. So we've had some interesting data, but there are a few things that we still need to do. Next steps are to explore the virus's safety and efficacy in non-human primates. We'd like to dissect the various contributions of the antibodies as well as the T cells in both developing immunity to immunization as well as its role in protection from disease. And now we're actually going to try to elucidate the mechanisms underlying immune induction of Aripo Zika in vertebrate cells. So with that, I'll be happy to take any questions.
Thank you very much. Super interesting talk and very promising. If you guys have any questions for Jonathan, could you please type those on the Q&A? I have one. Let's kick with one that I'm just reading. This comes from Ben. Have there been any documented cases of live attenuated viruses causing horizontal gene transfer, virus? How do you choose a virus genus as the backbone of your virus? So
That's a good question.
Two questions.
So, yeah, two questions. I'll start with the second one. So how do you choose a virus gene instead of backbone? So there are numerous viruses that you can see. Oh, did I put my video on? I don't want you to talk to a blank screen here. Okay.
Yeah, you're right.
All right.
Yeah.
So there are numerous viruses that you can select as a backbone. It does not necessarily even need to be a virus within the same family. So example, you can use an adenovirus to be a good vaccine vector against flaviviruses, against other alphaviruses. What you're really going for is something that would facilitate either the immune evasion, the immunogenicity that you wish. So these can be, pretty much implemented across the board. You can pretty much use any virus, really. It doesn't have to be specific to a particular pathogen. It really depends on whether or not that virus vector that you would like to use as your backbone is receptive to new gene sequences. So for example, let's say I wanted to put Ebola virus on Aripo virus. Odds are it is likely to happen, but some viruses might just not be would not be able to be rescued. So that's one of the limitations. And have there been any documented cases of live attenuated viruses?
And then he has a second question.
Yeah, the live attenuated viruses causing horizontal gene swap. Yeah, so I have not seen any evidence. I've not seen any studies that outline virus-to-virus horizontal gene transfer, but that is particularly interesting as well.
Yeah.
What range?
Okay, there's one more question. What range types of other viruses might this chimera virus approach work or not work?
Right. So Flavivirus chimeras have been around for a while. So there is broad applicability. Flavivirus chimeras using other platforms like dengue, using other backbones such as Japanese encephalitis, such as other backbones such as yellow fever virus, have been used to express a large number of proteins from other viruses, including things as far as Ebola, including other things such as alpha viruses. This virus, I would say, my virus, Aripo Zika, would likely be best suited for additional Flaviviruses because of its close evolutionary relationship and the fact that we were actually able to rescue a full virus. The other platforms like the yellow fever and Japanese encephalitis might be better suited because they're replication competent. So I would say that it can be applied to multiple other pathogens, but it will likely be best suited for Flaviviruses.
Okay, we have one more question. It comes from Rebecca Salgado. Great talk. Are the structural genes in your chimera based off the African or Asian strains of Zika virus?
So that's a good question. So our current construct is based on an Asian strain, an Asian American strain. It's based on the PRV ABC59 strain of Zika virus. We did challenge studies, which is probably what I think you might be going here. So it is based on American strain, Asian American, but we challenged with multiple strains of Zika virus throughout the time. I did not show all that data. But even though we used that Asian American strain, we can still show clear protection against other lineages of Zika virus, like MR766, which is West African, like the Dakar that we use and I showed here, which is again West African. Odds are there'll be actually better protection if we used more American strains for the challenge studies.
Thank you. Any other questions? Okay, well, if no more questions, thank you very much, Jonathan, for joining us today. It was super interesting and very promising, and I'm very happy to see this research. I assume the FDA is already on board with the preparation of phase 1 clinical trials and things like that. And if not, they should. But anyway, congratulations. It's a really great research. Okay, thank you very much, everybody, for joining us.
Thanks, everyone.
Take care.